Welcome to this talk on experimental design. My name is Carolin Moessnang and I'm happy to walk you through this very conceptual talk. And I want to start with the most important slide of this talk. Actually, the session on experimental design could be shifted to the very beginning of the entire course because the entire process starts with a good design. In fact, your research question directly translates into your experimental design, which means, of course, in the beginning, you have to think about the cognitive process you're interested in. And this cognitive process, in order to isolate this cognitive process, you have to think well about what kind of conditions do you want to have in your experiment? What would be a good control condition? How should those conditions be compared to each other? So this means that the experimental design will define what kind of research question you're going to be able to answer in the very end, how you will collect your data, how you will set up the entire experiment. Now, I also want to remind you about a very basic feature of fMRI. The bolt signal itself is not an absolute measure, but we always have to compare the strength of activation between conditions. It's a relative measure. So we have to keep in mind that in order to maximize the sensitivity of our experiment, we have to maximize the change in functional activation between conditions. That's pretty clear, I'm sure. A very simple example is that in one condition we have finger tapping, and of course the best would be then in a different condition which we want to have as a control condition is not to move the finger in order to isolate the brain regions which are supporting finger tapping. You know this figure very well. It's the processing hierarchy. And when you want to locate the experimental design in this hierarchy, you will find it in the design matrix. The experimental design directly translates into the design matrix. But as I said before, actually, we could shift this at the very beginning of your SPM processing hierarchy, because the experimental design will, in fact, define what kind of data you're going to collect at the very beginning. So here I want to give you an overview over the experimental designs offered by SPM. SPM, statistical parametric mapping, is using parametric statistical analysis, all within the framework of the general linear model. And those three designs here, categorical, parametric, and factorial designs, are all versions of the general linear model. They're all, in this respect, they're all the same. However, it is convenient to differentiate between those versions in terms of the experimental design. And this is also going to be the outline of my talk, where we first talk about the categorical designs, then about the parametric designs and the factorial designs at the very end. I want to here give you a very first overview. For categorical designs, we just compare two conditions, one of which contains the process of interest, for instance, finger tapping versus rest, and you are hopefully isolating those brain regions which are related, which are serving this specific cognitive process. In parametric designs, we rather look for a systematic relationship between brain activation and a specific process which we vary in its strength or intensity. So we ask the question, the more of the process, the more of brain activation. In this example, it is the change in brain activation with increasing finger tapping rate. Here you can see four representative subjects. For each of those four subjects, there are 3 voxels which are plotted, and you can see that the signal intensity increases with increasing finger tapping frequency, finger tapping rate. And finally, factorial designs are the most flexible designs. Here you can test for interaction effects, which is nothing else than you test whether the effect of a specific task changes as a function of context, whether, for instance, a specific context leads to a stronger or a less stronger effect, or to the activation in different parts of the brain. As an example here, again, it's about finger tapping. Here, the researchers were interested whether finger tapping activation related to finger tapping is different in younger versus older subjects. So here, the context is age, which can be tested for. You can do within subject or between subject contexts. Within subject would be, for instance, does the effect of finger tapping change as a function of the attentional state someone is in? We will now have a closer look at all the three different designs, and we'll start with the categorical design. And regarding the categorical design, we will start with a very basic idea of subtraction, cognitive subtraction, and we will look at those two watchouts here, the baseline challenge and pure insertion. So cognitive subtraction in itself is a very simple rationale. The aim is to isolate those brain regions which are supporting a specific process, cognitive process, which we here just call the process P. The procedure is as follows. You have your subjects engage in a task, in a condition which includes the process P, and you have them perform a control condition, which is identical, except for the fact that the subjects are not engaging in process P. When subtracting those two brain activation maps, you are left with the activation which can be attributed to your process P. An example is here. We have the experimental condition on the left side, the control condition here. You can see that they are perfectly matched in terms of lower level visual features, but not in terms of color. So when contrasting those two conditions, you're left with the functional areas supporting color perception. This is pretty simple, but there are two watchouts. One of which is you have to think well about a good control condition, and this is not trivial. So the first is the question of what is a good control condition, and you will see that this is not trivial at all. Let's now use this example here. Let's say we're interested in face recognition as our process of interest. So what would be a good control condition in order to isolate brain regions related to face recognition? So you could show your subjects a simple crosshair. Would this be a good control condition? Certainly not. While you will certainly have functional activation in brain regions related to face recognition, you will also end up with a lot of brain activation in different brain areas related to the processing of basic visual perception, but also related to mind wandering, since rest is never truly rest. You could also show a complex object as complex as your face you're showing in the experimental condition. Is this a good control condition? Well, again, you might not be able to isolate your processes of interest because you would have to control for the more basic processes related to face recognition, which would be face perception. So in these two examples, there are too many aspects or components in your control condition that are differing. So these are not good control tasks. So you could then think of showing a very similar stimulus in the control condition, also a face. And in this experiment, you might show them a non-famous face, which cannot be recognized. Well, the problem here is that subjects might start to wonder, oh, should I know that person? Is it maybe famous? And with this, implicitly engage in the process of interest. Also, it might turn out that the control condition will be then more demanding than the experimental condition, which is also then not a good match. Also, what you could do is a very elegant way actually is to show the very same stimulus, but have the subjects engage in different tasks. So with this, you perfectly match the visual input. And you could, for instance, ask your subjects to name that person in the one condition, and in the control condition, name the gender, where the recognition itself is not necessary. But of course, also here, subjects might engage in face recognition in the control condition as well. And also, there might be a problem of showing the same stimulus twice. Seeing the same stimulus a second time might lead to changes in the functional activation, which we call interaction effect, interaction with time, which I come to in a few slides. So I hope with these examples it became clear that depending on the choice of a control condition, you will answer very different research questions, and that finding the correct baseline is not trivial at all. Now let's move on to the second watch-out, which is pure insertion. Pure insertion means that adding one component does not affect the other components. And I brought you an illustration with latte macchiato. On the left side, you can see a perfect latte macchiato where the insertion of coffee almost did not affect the milk. Whereas in the second latte macchiato, the insertion of coffee had an effect on the milk. And you can imagine that in biological systems, there is a lot of interaction going on between processes. So the interaction or the non-additivity of processes is very common. Put it in different words, pure insertion is rather the exception than the rule in the brain. Now let's look at this in the brain. Let's assume we have a control condition where subjects engage in a task which does not include process P, and then we include process P in the experimental condition. And you can expect that this inclusion of process P will also affect the other processes. You can see here that the brain activation changes just by including process P. And when you do the subtraction, you're left not only with activation related to process P, but also with those interaction effects. And in fact, this is the problem of cognitive subtraction, or the great disadvantage. It depends, it rests on the assumption of pure insertion and assumes that we have an additive effect. But in the brain, it's mostly not the case that processes are additive. We rather end up with nonlinear effects, that the combination of two processes, for instance, lead to even more activation than expected, or the opposite, that we are left with less activation than expected. So we have nonlinear effects. And I want to give you an example for a nonlinearity in the processes, in the neural processes. You remember the idea of a control condition where you just use the very same stimulus twice and you have subjects engage in different tasks. Well, what can happen, or which has been shown, what happens when you show the same stimulus twice is that you have an adaptation effect. You see in the first presentation, you have a stronger effect than when the brain is presented the same stimulus a second time. So you have a reduction in the activation. And of course, if you don't take this into account, you might wrongly assume that the difference in brain activation is due to the difference in task, while actually it is an effect of time. We have a task by time interaction effect, which needs to be accounted for. So how can this be accounted for? The problem of the control condition, the problem of pure insertion, and one elegant way to address this is conjunctions, which we're going to look at next. So to understand conjunction, let's go back to this depiction where we are left not only with process P, but also with activation resulting from interaction effects in one contrast one. Now let's assume we have a second contrast where we have subjects again engage in a specific task, including process P and a control condition. It's different, but it has the same rationale and it includes the same process P. So in the contrast, we are left with functional activation related to contrast P, but also with other interaction effects. And now you can already see what's the beauty of conjunction. We have a third step where we look at the overlap in brain activation between those two contrast images. The idea here is that when we look for the overlap, all those interaction effects we're not interested in are just cancelled out. So here we can very elegantly address the problem of pure insertion. So this is a schematic depiction of the cognitive subtraction and conjunction rationale. In the cognitive subtraction here on the left side, let's assume we have two tasks, task A and B, and we have 5 processes. Those two tasks are perfectly matched except for the process number 4, which is our process of interest, PI. Again, we know that pure insertion is the exception rather than the rule. We can expect interaction effects, and we're not happy with that situation, so we opt for a conjunction analysis. Here again, we have one task pair, but also a second task pair. Both task pairs include the contrast, the process 4, which is our process of interest. Now, in the task pair 1, which are the conditions A and B, those two conditions are perfectly matched with regards to process 1. They're not matched with regard to process 2. There is no process 3 involved. They're not matched on our process of interest, of course, and they're perfectly matched with regards to a certain process number 5. So when we do the cognitive subtraction here, we're left with effects of process 2 and process 4. In the second task pair, condition A does not include process 1. It does include process 2, the same for the control task B. Then we have process 3, which is present in the condition A, but not in the condition B. We have process 4, which is present in condition A, but not in condition B, and there is no process number 5. So when we do the subtraction here, we are left with activation related to process 3 and process 4, which is the ones of interest. So when we compare those two contrast images, the only thing which is overlapping is the contrast in process number 4. And this leaves us those brain regions that are specifically engaged by process number 4. In other words, with conjunction, we can isolate the context invariant activation which are associated with a specific cognitive process. I want to illustrate the conjunction rationale with this example. So it's about a study where researchers were interested in the neural structure supporting phonological retrieval. For instance, here you see a dice. And in order to be able to say this is a dice, you have several processes, which is visual analysis of the object, the recognition of the object, then the phonological retrieval, and then in the end, the verbal output. The phonological retrieval is the process which is of interest in this study. So in order to isolate the brain regions that support phonological retrieval, the researchers realized 4 conditions. In this condition, subjects had to name a word, read it out. In the second condition, they had to name a letter. In the third condition, they had to name an object, and in the fourth condition, they had to name a color. Each of these conditions also had its control task, which was just saying yes, independent of what they see here. So in this control condition, it was already a control for the verbal output. The process of verbal output was controlled for. Those 4 conditions were very different in terms of processes. For instance, in the condition words, in order to read out a word, there are different processes involved, like form processing, lexical orthography, semantics, processing of semantics, and finally phonological retrieval. And each of those conditions had different processes. The only process that they all had in common was the process of phonological retrieval. So the idea is now to do a conjunction analysis in order to isolate the brain regions supporting phonological retrieval independent of the item, independent of what has been processed in terms of those conditions. So, and this is the result. This is the conjunction of those 4 single contrast images and you can see it is involving temporal, occipitotemporal structures, but also frontal structures. You see phonological retrieval in its context invariant realization is involving several parts of the brain. Of course, there might be still interactions of processes which we did not account for in this experiment, but at least those which have been addressed explicitly can be ruled out. Now let's move on to the parametric designs. And we will start with linear and nonlinear parametric modeling. So in parametric designs, what we do is that we vary some features of a stimulus systematically on a continuum, and we relate the bold response to this variation. We can test for linear effects. For instance, we can ask whether there's a linear increase in functional activation in parts of the auditory system with increasing loudness of the stimulus. We can also test for nonlinear effects, which is oftentimes the case. We can also relate readouts of an experiment, for instance, response times or certain parameters which we are computing based on behavioral data to the brain response. So we can use behavioral readouts, for instance, to ask test for associations with our functional activation. One pretty thing about parametric designs is that we are avoiding the pure insertion problem because there is no new process P we're including. There's just an increase or a decrease in the intensity of the process. We also have no baseline problem because the lower realizations of the stimulus feature are at the same time also the control for higher realizations. It means that we're testing for relative changes anyways, and this is already realized in varying our stimulus characteristics on the continuum. So this is a very simple example. In the auditory cortex, words were presented at different rates, and you can see that in the left and in the right primary auditory cortex, the functional activation increases with increasing presentation rates of the presented words. So the previous example was a linear relationship. You can also test for nonlinear relationships. These are called polynomial expansions of these associations, and this is implemented in SPM in a very convenient way. So this is an example of a nonlinear effect, while this is a single patient which had a stroke. And again, this patient was presented words at different rates. And you can see in the auditory cortex, as before, there's a linear relationship with increasing word rate, while when they were modeling a quadratic relationship, they found an effect in the prefrontal cortex. This effect would not be detected if you would not test explicitly for these higher order polynomial terms. Now I also want to give you an example for a model-based regressor. So this is an example of a study which used such model-based regressors. In this study, subjects had to learn specific probabilistic outcomes. They, for instance, here saw specific stimulus, and over the course of the experiment, they learned that when pressing the left button, they had a 70% chance to win 25 cents. When pressing the right button, they had a 70% chance to only win 10 cents. And there's also a lower chance, but also a chance that they would not win anything. So over the course of the experiment, subjects learned predictions about obtaining a reward. And what can be now modeled based on those behavioral data is a so-called prediction error, which means the difference between the expected outcome and the actual outcome. You can also read it as a surprise signal. So for instance, on the third trial, subjects were more surprised than on the second trial, so the outcome was not as expected, and so on. So there is a prediction error signal that fluctuates across trials, and now you can ask which brain regions do relate to this prediction error signal. And as expected, researchers found the strongest association of this prediction error in the ventral striatum, which is associated with reward processing. Now, having learned about the beauty of parametric designs, I want to show you also the most flexible way of designing an experiment, which is the factorial design. Already here, I want to point out that you can combine parametric and factorial designs, but we will come to this in a second. So first, a quick reminder about what is a factorial design. Now here you can see an example of a 2x2 factorial design. You have two factors, and each of the two factors has two different realizations. Let's say we have one factor is age, you have adults and kids. One factor is sex, you have male and female. And in a full factorial design, you have each of those combinations realized. The nice thing about factorial designs is that it's highly efficient. Why that? In fact, you can test explicitly for interaction effects. And you remember, interaction effects have been the problem of the cognitive subtraction. And you can use, for instance, conjunction analysis to erase those interaction effects. In factorial designs, it's not erasing those effects. No, you explicitly model them, and you can have a look at those interaction effects, which is, of course, an even smarter way how to deal with interaction effects. For illustration, let's go back to the experiment on phonological retrieval. This time, the question is whether phonological retrieval, what areas support phonological retrieval in the context of object recognition. So the question the researchers were asking is whether the inferior temporal cortex is sensitive to both object recognition and phonological retrieval. And they started off with three conditions only. The first condition was an abstract image. They had to say just yes. So here we have the cognitive processes of visual analysis and verbal output. In the second condition, they should just say yes when they see an object. So here we have the visual analysis. We have, in addition, object recognition, and we have the verbal output. And finally, in condition C, they had to name the object. So it's not only visual analysis and object recognition, but also phonological retrieval, which here now is of importance, and again, the verbal output. And the hypothesis of the researchers was that this specific cortical area would not only be sensitive to object recognition, but also to naming an object, phonological retrieval of objects. So they were also assuming an increase in activation from condition B to condition C. Now, this is the result. When they plotted the bold response in this area, they would see an increase in activation between just seeing an abstract image versus seeing an object or recognizing an object, but there was no difference in activation between object recognition and naming the object. So falsely, one could conclude, okay, this brain region is not involved in phonological retrieval of objects. However, the problem is that there could be an interaction effect. So you remember that slide. It could be that we don't have this scenario here when adding object phonological retrieval regarding objects and object recognition. There might not just be an additive effect, but we might have such an effect that the combination of both somehow leads to a nonlinear change in activation. In order to test this, we need to explicitly address interaction effects. And how can this be done? You have to be able to vary both factors, phonological retrieval and object recognition, independently from each other. And for this, we have to have a fourth condition. So here you again have the three conditions we just walked through. Now they added the fourth condition, which is naming a color. Here, with color naming, you also have phonological retrieval, however, not in the context of object recognition, because color is no object, obviously. Now you have a full factorial design. So now let's look at the interaction effect. And you can see here that the difference in activation within this inferior temporal cortex is sensitive to object recognition, dependent on the absence or the presence of phonological retrieval. In this condition where we just have to say yes, the inferior temporal cortex does make a difference between objects and non-objects that you can see here. However, in the presence of phonological retrieval, this is much stronger. There's even a deactivation when we have phonological retrieval when there is no object. So it is sensitive to both contexts. This effect wouldn't have been seen if we would not have addressed the interaction effects explicitly. Now let's look at parametric effects within the factorial design model, which means that we can explicitly test for linear and nonlinear interaction effects using parametric regressors. So this is an example. Again, it's about word rates. This time subjects were asked to produce words. They had to produce words either by repeating words, or they had to produce new words. And they had to do this at different frequencies and at different rates. So the parametric condition here is the frequency with which those words were generated. And the categorical manipulation was whether it was about repetition or the production of new words. And one could assume either a main effect of condition that, for instance, the production of new words would always lead to an increased activation in specific areas, while the repetition of words would be always at a lower level, but still we would have an increase with increasing production rate. Or one could have also an interaction effect that the increase in brain activation would be much stronger, for instance, for new words compared to the repetition. And what has been found is indeed an interaction effect in the PSTS region, where the increasing frequency of repeating words led to an increase, while it led to a decrease when it was in the context of producing new ones. So this is just an example for an interaction effect with parametric regressors. We ask whether the effect of a parametric variable is different dependent on the specific context. And finally, I want to come to a very specific form of a parametric modulation in the context of different contexts. It's the psychophysiological interaction. Psychophysiological interaction, or PPI, is a measure of functional connectivity. And it is actually asking whether the functional connectivity between two brain regions is dependent on specific context effects. An example is the communication between two brain regions, or the functional connectivity between two brain regions as a function of attention. Let's assume that only in a high attention state, two brain regions, as for instance here, R1 and R5, are highly interconnected, highly synchronized, whereas in states of low attention, they are not. Such research questions can be answered using psychophysiological interaction. So in order to calculate PPI, I want to give you a very quick overview. I highly recommend the paper by O'Reilly and colleagues. I also used the figure here from that publication. In order to calculate PPI, we need the information about the task, but we also need information about the time course in a specific region of interest, our seed region. And we want to know what other areas in the brain are increasing their connectivity to this specific seed region as a function of task. So we need, of course, the task regressor. Let's say here the finger tapping task, we have periods of rest and periods of finger tapping. After convolution, we have a task regressor, which we're feeding into the experimental model. Then we have we need the regressor of a seed region. Let's say we are interested in our hand knob region and we want to know which areas of the brain have an increased connectivity with the hand knob region explicitly and only during the finger tapping, but not during rest. For this, we have to extract the brain, the bolt signal from the hand knob region, shown here in blue. We have to deconvolve it, and what we now have to do is to build the PPI regressor, which is an element by element product, like a multiplication of the task regressor, here in black, and the seed region, the bolt signal in the seed region in blue. When we do this correlation, we will end up with a red regressor here. The red regressor is the product, and you can see that this red regressor shows an anti-correlation to our seed region's regressor during the periods of rest, while it shows a high correlation with our seed region during the task, during finger tapping. So this PPI regressor makes sure that when we feed it into the model, we will only isolate those brain regions which are highly synchronized to our seed region during the periods of task of finger tapping, but not during rest. And with this, we can specifically address the effect of context. So with this, I want to close this session. I hope you got a good overview over all the different designs, that you keep in mind the watch-outs, that you think about the beauty of the factorial design also combined with the parametric design. Good luck with the all analysis to come, and thank you for listening.